Hello everyone and welcome back to AP Bio with AP Playground. Today we're reviewing Unit 4, Cell Communication and Cell Cycle. Things you need to know for this unit: the signal transduction pathway, as well as all of the components of it including receptors, signaling pathways, and responses, how changes of the pathway can affect the response or the pathway, positive and negative feedback in maintaining homeostasis, the cell cycle, specific steps of mitosis, and regulation of the cell cycle. Let's get into it. First up, 4.1, types of cell signaling. So there are four main types of cell signaling, and it's not necessarily needed to know all of these types, but it's good to be able to recognize different examples when given problems related to cell signaling. So first there's autocrine, which is where the chemical signal, otherwise known as the ligand, is going to affect the sender. So it's essentially self-signaling, and you can see it right here. There's also juxtacrine, which is signals that travel by direct cell contact, and these in plant cells occur from plasmodesmata, which are the pores in the cell wall, and in animal cells through gap junctions. That can be seen right up here with gap junctions and plasmodesmata. And one way this is used throughout the body is in the immune system, and the helper T cells are trained through contact, as well as killer T cells, which kill infected cells through contact. So contact is especially important when talking about the immune system, because it's how our immune system trains to recognize which cells are friendly and which are foes, and then killing the unfriendly cells. Then there's paracrine signaling, and this is short distance and to coordinate with neighbors. And this can be done within an organism between different cells, not necessarily right next to each other. It can be done between multiple organisms in the sense of quorum sensing, where bacteria are determining the population density of the species by sending out a signal, and depending on how many other members of the population there are in that area, the bacteria will then pick a course of action that prevents it from taking resources away from the other members of the population. And that's how it's very important in coordinating and communicating with neighbors. This happens also in our bodies through synaptic signaling right here with neurotransmitters, which are how neural signals are transmitted, and neurotransmitters are released between neurons through paracrine signaling. And then lastly, this is kind of the different one from the three prior that we discussed, and that's endocrine signaling. This is long distance, and the way signals and signaling molecules travel long distance is through the bloodstream. And the majority of the molecules that travel along the endocrine pathway are hormones, which are released in various places of the body, but then do act in other places, or the signals to start production of those hormones in the pituitary gland then get transferred down to wherever those hormones are produced. And some important hormones that we have to consider are obviously the ones that get a huge spike during puberty with estrogen and testosterone, but also insulin, which is going to regulate blood glucose level, and College Board loves to use insulin and the insulin pathway as an example of a cell signaling pathway. So we'll be taking a look at that later. Now it's time to talk signal transduction pathway. And these are the stages in which cells receive, amplify, and respond to a chemical signal known as a ligand. And ligands can only bind to specific target cells because those are the target cells which produce the receptors that take in the ligand shape. So, like I said earlier, three components of signal transduction: reception, where there's a very specific binding, just like a substrate to an enzyme, there's a very specific binding of ligand to receptor. And then we have transduction, where the signal is amplified and essentially translated to the point where it can create a cellular response that can either be a metabolic process or a change in what proteins are being produced by the cell. And we'll talk more about each of these steps in depth right now. So, part one of signal transduction pathway: reception. Now, reception can either happen inside the cell or on the cell surface. And inside the cell is going to be through steroid hormones, which again are transferred through endocrine signaling. And the reason for this is that for a molecule, signaling molecule, ligand, to make it inside the cell, it has to pass through the cell membrane. And it needs to be a molecule that does not have a charge, that is small, so it can pass through the cell membrane without extra assistance. And that's why steroids are what are making it through the cell membrane, because they're hydrophobic molecules and they're small as well. So, and then this kind of reception is generally going to lead to gene expression, where a certain part of the genome is now going to start being acted upon, and some proteins are going to be produced. Now, cell surface receptors, there are a few kinds, and the general idea is that the ligand is going to be binding outside of the cell to the receptor, but the receptor also has an intracellular domain that's going to allow the signal to then start the transduction process to a cellular response. So there's ligand-gated ion channels where the ligand attaches, the gate is going to open or close, and that's going to allow ions to pass through, as you can see right here. And this is how neurotransmitter and synaptic signaling works. And when ions pass through, that's going to change the polarity of the cell. There's going to be, because ions are either positive or negative ions. So there's going to be a change there, and that can have effects later on in transduction. Now, there are also G protein coupled receptors where the signal is transmitted through the G protein. When the ligand binds to the receptor, as you can see right here, there's going to be an activation of the G protein and through the use of GTP, and then that G protein is going to enter an enzyme that will then start the transduction process. So there are a few steps to a G protein coupled receptor, but the idea is the same. There's a ligand that's entering the receptor, and that's going to start the transduction process. And lastly, there are receptor tyrosine kinases, and these are just enzyme-linked receptors that are going to transfer phosphate groups to tyrosine, and that's going to bind to growth factors. So just like a G protein coupled receptor, when the ligand binds, the receptor is linked to an enzyme that's going to then become activated once the receptor binds. So in general, there can be intracellular or cell surface reception, and they're all going to be from the ligand binding to a reception molecule and beginning the amplification and transduction of the signal. Now, what actually is the transduction step of the pathway? How does the signal become transferred from the receptor all the way to creation of a cellular response or reaching the nucleus? And this is going to happen through amplification. That can happen generally one of two ways, or a combination of both. The first one is a phosphorylation cascade, and it is what it sounds like. There's going to be a cascade of phosphorylation, or the addition of phosphate groups. And it's one way to activate proteins in the chain, and that's through the addition of phosphate groups, because that's going to add negativity to each protein. So there's going to be a transfer of phosphate groups that's catalyzed by the enzyme kinase, and then later, once this process needs to repeat, the phosphate groups are going to be removed by phosphatases. So as you can see, there's as the signal transduces along the pathway, there's going to be protein kinases that are going to become activated through the transfer of a phosphate group from ATP, until eventually we lead to a cellular response. Now, second messengers are non-protein molecules that pass along the signal. So compared to phosphorylation cascade, which is based on enzymes that are being activated through phosphate groups of ATP, these second messengers are non-proteins. And the two most common kinds are calcium ions and cyclic AMP. And calcium ions act as a good second messenger because there's not many in the cell. So when they're able to enter the cell through, say, a ligand-gated ion channel, then there's going to be a huge shift in negativity, or in positivity in the cell, because these are positive ions. And proteins often have binding sites for the ions, which are going to change their shape. And cyclic adenosine monophosphate, cyclic AMP or CAMP, is helpful in activating a protein kinase, which is going to then start the phosphorylation cascade. So second messengers generally work in tandem with a phosphorylation cascade to assist it or to begin it, so that then the signal becomes transduced. Now, there are two kind of main responses to signal transduction. The first one is gene expression. This is where the signal is reaching the nucleus, and now some DNA is going to be activated, and protein synthesis. There's going to be some proteins that are going to begin created, or there's going to be the stopping of production of proteins. And changes in the environment resulting in signal transduction can alter the phenotype of an organism, or the outward appearance, because there's going to be a change in the proteins that are produced. Or there can be a metabolic response. So there's not going to be a change in protein production or gene expression, but there is going to be an effect of the protein activity already in the cell, like enzymes. There's going to be either you can turn off a certain enzymatic pathway, or then you can contribute to it. Also, there's this response known as apoptosis, which is programmed cell death, which is a very systemic suicide of the cell and prevents damage to neighboring cells, because this cell that is old or broken is not helping the organism, and by apoptosis, this programmed cell death, its resources can be used and given to other cells, and it's not going to be a problem in the body. And this is also what's going to be helpful in the creation of our hands and feet and separating each of our fingers and toes. Now, one of the common pathways is the epinephrine pathway, also known as the adrenaline pathway. And when epinephrine, which is a hormone, is released, there's going to be a breakdown of glycogen to increase blood glucose as part of the fight or flight response. So we can see the epinephrine pathway here, and we can take a look at what the response to the signal transduction is. So epinephrine is going to enter the receptor, and you see we have a G protein coupled receptor here that's then going to activate adenylyl cyclase, which is producing cyclic AMP, our second messenger, and that's starting our phosphorylation cascade that's eventually going to lead to the release of glycogen from the cell, and that glycogen is going to be broken down, and now we have extra blood glucose in order to either fight or fly. Now, it's important to remember that a lot of the components of the signal transduction pathway are going to be proteins, including the receptors and the kinases in the phosphorylation cascade. And when we have a too high or low pH or too high or low temperature, the protein can be denatured because we're not in the optimal zone anymore. And this is going to obviously affect the activity of these components of the transduction pathway. Additionally, mutations in genes can affect the proteins of the pathway because a mutation is a change in the DNA sequence, and if the DNA is altered, then the protein synthesis and production of these receptors and kinases are going to be changed. Either we're producing now a different protein or not at all. And we'll talk more about mutations in Unit 6. Lastly, a lot of components of the signal transduction pathway, like the kinases we looked up in the phosphorylation cascade, are enzymes. And enzymes can be inhibited or activated through the active site or the allosteric site. And this through competitive or non-competitive inhibition that we talked about in Unit 3. So not only can the gene expression be altered, but also the metabolic processes of these enzymes can be altered, which can all affect the signal transduction pathway. Now it's time to talk about signal transduction in the context of feedback loops. So homeostasis is the tendency to resist change in order to maintain a steady and stable environment. Essentially, we're always trying to return to our set value. And negative feedback loops are going to oppose the stimulus. We're going against a change to get back into our set value, back to homeostasis. And most systems have two negative feedback loops. One if the stimulus goes too high, and one if it goes too low. And this is negative feedback. So we can look here at the insulin and glucagon feedback loop. When you've got high blood sugar, that's promoting the release of insulin, which is then going to stimulate glucose uptake from the blood, and that's going to be stored as glycogen, and that's lowering your blood sugar. So we're returning back to the set value when the blood sugar is too high. Versus when you have a low blood sugar, then we're promoting the release of glucagon, which is then going to break down glycogen and increase blood sugar. So there are two negative feedback loops. And negative feedback does not mean that that we're necessarily trying to lower something. It can be that we're trying to raise a value. We're just trying to get back to our set value. Now, positive feedback loops are going to amplify the signal, and they're moving away from the set point. And some examples here are going to be the onset of labor in childbirth, once there's those initial signals that the child is ready to be birthed, these signals are going to continue and keep increasing in strength until labor is complete. And then also there's ripening a fruit through the use of ethylene. So we can see here in this pathway, an apple on a tree ripens, and this ripe apple produces ethylene. Ethylene is going to then signal to the neighbors that they need to ripen. And then the neighbors are going to start ripening and produce ethylene. And then more apples ripen. And then that's going to cause more production of ethylene. And then the neighbors are going to start producing ethylene, and more apples are ripened, and that's going to lead to the production of more ethylene. So positive feedback loops we're moving away from the set point. We're amplifying the signal and causing it to spread. Whereas negative feedback loops, we're returning back to the set point. We're done with signal transduction. Now it's time to talk cell cycle. And cell cycle is the life of a cell from formation, from when it divides from a parent cell until it divides itself. And 90% of the cell cycle, which isn't going to involve dividing, is known as interphase. And this is the period between division. And interphase is broken up into 3 parts. Number 1 is G1 phase, or growth 1, or gap 1, where the cell grows and it's going to carry out its cellular function. Then during S phase, there's going to be a duplication of chromosomes. And then during G2 phase, there's going to be continued growth. And why are we duplicating chromosomes? Because we need, in order to have the cell divide and create 2 identical cells, it needs to have double the amount of genetic material, or genome. And each chromosome is going to become 2 sister chromatids attached by a centromere. So really the number of chromosomes is the same, but now each chromosome is going to have a sister chromatid. And we can look here at the human genome, which has 23 pairs of chromosomes, or 46 in total. And if we look at pair 1, for instance, this is a pair of homologous chromosomes, pair 1. So there's going to be one of these came from the mother and one of these came from the father. And we can see here the homologous chromosomes, and at S phase, when these chromosomes replicate, now each homologous chromosome is made up of 4 sister chromatids. We have sister chromatids on the parent chromosome and sister chromatids on the other parent chromosome, on the mother and the father chromosome. And then during cell division, these chromatids are going to be pulled apart so that in the resulting cell, each one is going to have a chromatid from the father and a chromatid from the mother. We're creating identical cells from the parent. So the last 10% of the cell cycle is going to be mitosis, which is that division of the cell's nucleus, and then followed by cytokinesis, which is division of the cell's cytoplasm. And during growth 1 and growth 2, we're getting ready to create this division. So we're duplicating all of the organelles, and during synthesis, we're duplicating the genome so that the nucleus can divide. And mitosis is involved in growth because as you're dividing cells, then now you've got more cells and you're growing. You're repairing tissue in case some cells are damaged, and asexual reproduction, which is the reproduction in which you're cloning yourself. There is no genetic variety. And mitosis is producing clones, exactly identical cells. And in prokaryotes, such as bacteria, the cell division happens through binary fission, which is very simple because prokaryotes only have 1 strand of DNA which duplicates, and then the cell pinches and divides. Now let's talk about the 4 steps of mitosis in cells, and these are in eukaryotic cells, and these can be remembered by the acronym of PMAT. First is prophase. This is where the chromatin in the cell, the genetic information, is going to condense into chromosomes that are going to be our, very solid and linear kind that we looked at in the last slide. And the nucleus is the nuclear envelope is going to fragment and break apart, and the mitotic spindle is going to form, which is going to start creating microtubules, which you can sort of see here, that are going to then attach to the kinetochores, or the center of all of our sister chromatids. And when we reach stage of metaphase, some books include prometaphase as an in-between stage where this continues. When we reach metaphase, all chromosomes are lined up in the very center of the cell at a metaphase plate, and all of the chromatids have been attached to by microtubules from the mitotic spindle. And then when we get into anaphase, these sister chromatids are going to be separated by the microtubules, and the microtubules are going to start pulling them to opposite sides of the cell. And then lastly, in telophase, these nuclei are going to reform, and then in cytokinesis, we have the division of these two cells, which happens in animal cells through a cleavage furrow, which kind of pinches at the center until the cell split. And in a plant cell, because there's a cell wall and there needs to be a formation of a new cell wall for each of these quote-unquote daughter cells, there's a cell plate that forms that then becomes the new cell wall. So mitosis is going to be division of the nucleus, and then cytokinesis is division of the rest of the cell. And mitosis happens by lining up all of our sister chromatids in the middle of the cell and then pulling them apart through the use of microtubules. And from this point, each daughter cell is going to have the same amount of chromosomes as the initial starting parent cell, because remember, in S phase, we duplicated the chromosome, so it consisted of 2 sister chromatids. Now, what goes into regulating this cell cycle? There are certain checkpoints during the cell cycle that check whether certain conditions are met before the next stage can occur. Essentially, there are sort of guards at a gate saying you can pass or you can't. And there's one after G1, which is checking, does our DNA have mutations? Because we don't want to replicate, we don't want to go into S phase and replicate mutated DNA. Can the cell keep growing, and does it have enough energy to continue in the cycle? Essentially, do we have enough space to continue growing so that then later we can divide? Then there's a G2 phase checkpoint after synthesis. Has all the DNA been replicated properly? Because again, we don't want to divide with genetic information that isn't right. And then lastly, there's an M phase during mitosis checkpoint after metaphase that checks, have all the chromatids been attached to the microtubules properly? Because if some chromatids haven't been attached and then the cell begins to divide, then now we have more chromatids in one daughter cell and less in the other. And if the G1 checkpoint is failed, then the cell gets sent to what's known as G0, where it's no longer moving through the cycle. It's essentially left this process here. And some cells can leave G0 and continue through the cell cycle when necessary, such as liver cells, while others remain permanently in G0, such as our muscle and neural cells. So when you work out and your muscles grow, that's not because the muscle cells are dividing. The muscle cells are just getting larger because muscle cells stay in G0. And there are two more factors that are usually considered in the cell cycle, and one is density dependence, meaning are there too many cells in one area? Because if there are too many cells here and these cells start to divide, then there's not going to be enough resources for all of those cells. So generally, cells don't divide when there's so many of them near each other. And then lastly, anchorage, where is the cell attached to something? Does it have a base? And these checkpoints are controlled by the CDK cycle, or the cyclin-dependent kinases. And these are going to be active only when connected to cyclin proteins. If growth factors are present, that's going to contribute to the presence of cyclin, and growth factors mean that there's enough nutrients so cells nearby can also divide. If we're not too crowded, then we contribute to cyclin and we can pass through the checkpoints. If we are anchored, then we contribute to cyclin, we pass through the checkpoints. And obviously, those criteria that we talked about in the last slide to pass all of these checkpoints are going to contribute to the amount of cyclin present that's going to allow us to continue through the cell cycle. Now, cells which have no limits on division are not dependent on density. They don't care about anchorage. They're going to divide even if there is mutation. And as a result of this, they're going to steal resources from other cells to divide, are known as cancer cells. And a tumor is a mass of abnormal cells within normal tissue. And the tumor can be benign or malignant. Benign meaning it's not going to spread, while a malignant tumor will spread. And when it travels through the body, that's known as metastasis. And essentially, these cancerous cells are not going to be limited in their division, which allows them to take resources from other cells and generally leave people in a bad place. And that's it for Unit 4, and we'll see you in Unit 5 with heredity.